Good afternoon to those of you here in the classroom and to those of you online. My name is Bill Wells. I'm a member of the Science, Medicine, and Technology Subcommittee. And it's my job to introduce the folks that will be presenting today. The subject of this discussion is the latest research in bird population decline. And the National Science Foundation has awarded a $500,000 grant to two Hope College professors, Dr. Kelly Rowland and Natalia Gonzalez-Pech, sitting in the back seat there. And she's going to, if Kelly says anything wrong, she's going to correct her. So, and they have been studying the nanoparticles and how it may be affecting bird population. And I was stunned when I read that connection. I'd never heard of such a thing before. So I'm dying to know what the details are here. Kelly Rowland is a sensory psychologist, ecologist who joined Hope's Department of Biology in 2019. Previously, she was a postdoctoral researcher at Indiana University. Ronald received a BA from Transylvania University in Lexington, Kentucky, and a PhD in biological sciences from Purdue University. And I'm sorry I don't have your credentials in front of me, but she's almost international in her academic activities. So without further ado, it's yours.
Thank you all for having me here today. You heard a little bit of my career trajectory. You heard maybe that I was graduated from Transylvania University. That's another liberal arts school. And that's really where my interest in really big questions developed. So a lot of my research interests are across different disciplines. My collaborator here, Natalia, is in chemistry, so I really enjoy those big questions. I'm going to start today by showing a little film from the Cornell ornithology group here that introduces this topic.
Birds are part of our lives. They fill the air with song, inspire us with their beauty, and make their homes in the most unexpected places. Sometimes they're even our neighbors. But the birds we love are vanishing. An alarming new study reveals that the population of North American birds has dropped nearly 30% since 1970. That's almost 3 billion birds gone, vanished from our forests, grasslands, and backyards in less than the span of a human lifetime. Some of the hardest hit are familiar birds: orioles, meadowlarks, swallows, warblers. What's driving this decline? Birds are losing the habitats they need, places to live, find food, rest, and raise their young. They face many other threats as well, from free-roaming cats and collisions with glass to toxic pesticides and insect declines. Climate change will compound all of these problems and accelerate the loss of habitats birds need. This enormous loss of birds signals a broader crisis in the natural world, one that ultimately affects us all. But it's not too late. Our actions on behalf of birds make a positive difference and benefit the entire planet. Thanks to strong conservation efforts and habitat management, many waterfowl and formerly endangered species are now flourishing. It's time to expand these conservation efforts to help the rest of our birds recover too. With our help, they can be restored as a vital part of the American landscape and a precious part of our lives. We're united in pursuing better protections and increased support for birds. And you can help. Please join us.
Thank you. They just do such a better job than what I can convey in those 3 minutes, right? It leaves a lasting impression. When I was starting at Hope College, this paper came out that this video cites, this Rosenthal in 2019, right? So this number was staggering to me, 30% reduction in population size across species that are what we consider common, right? In particular, these American sparrows are the largest hit group, right? And so as I was designing what is the trajectory that I want my lab research to go while I'm here at Hope, this paper stood out to me. It was something that I was considering quite heavily about what species I was going to target with my work, what I was planning on studying, and how my work could be construed to the public and to my students, and how to get people involved in caring, right? So work from my lab has been picked up by both internal sources. So this was a little news article that was published about my lab's work in the News from Hope College, and as well as external work with my colleague here today. And so all of the work that the lab focuses on is really with this idea to help increase bird populations flourishing. And so there are a couple of different ways that we can study this and try to help mitigate this issue. Those two ways, or the two areas that contribute to bird populations flourishing, are first, the animals have to survive, right? So they have to be able to find food, and they have to be able to avoid predators. I saw in that video, house cats tend to be the largest threat to our passerine species, our songbirds that we know of. But in addition to surviving, they also have to be able to reproduce, right? They have to be able to find potential mates, create a nest, raise those offspring, typically with biparental care, and have that nest successfully fledge. And these two things are not separable, right? So as soon as there's offspring, they also then have to survive. They have to avoid predation, and they have to be able to rely on parents for food. So today I will be talking about my or our work with the NSF grant, but I also want to talk a little bit about where my work began, which was really on this second part, understanding female mate choice, because that contributes quite a bit to animals being able to find a mate and produce offspring successfully. Okay, so both processes require that the animal is able to detect and respond appropriately to environmental stimuli. I'll pause here and say that I'm a professor and I'm used to having questions raised throughout my talk, so don't feel like you're going to interrupt me. If you have a question right then and there, feel free to raise your hand. I'm fine with having a dialogue.
Please remember to wait for the microphone so that the people at home on Zoom can hear as well. Thank you.
Thanks. Okay, so I consider myself a sensory ecologist, okay? And that bridges the fields of animal physiology and animal behavior. So understanding the mechanisms of how animals are able to detect and respond to stimuli, and how that ends up affecting their behavioral decisions, finding food, avoiding predators, and finding mates. Okay, so sensory ecology really lies at the intersection of these two fields. Right, and a great way to study questions within sensory ecology is within the framework of animal communication. And within animal communication, we have a sender and a receiver. They're within a given environment. With this species here, the house sparrow can be in an urban or a rural environment. That sender has to communicate in a variety of ways, perhaps through the use of song, perhaps through the use of visual displays, to that receiver. That receiver has to be able to detect that signal or signals and then respond appropriately. So this is the framework that the lab tends to ask all their questions in understanding this communication process and what happens if we change any one of these components and how that might affect how the bird is able to respond. So the major outlines of my talk are, advance forward. Firstly is, how does female sensory physiology affect her preferences for a mate choice display, tying in with that reproduction aspect. And then I'll end with, how does environmental change impact animal communication? This is really where the lab is right now. The first bullet point was more my dissertation work. Okay, so courtship signals are incredibly complex, right? And can span many, many modalities, right? So here's just a few. I've removed the sound on these, but our sage grouse is producing a sound at the same time that they're making this visual display. This peacock spider, aptly given its name, is sending vibrational signals through that substrate at the same time it's waving those arms. Much like how you're paying attention to the sound of my voice, you're also looking at how I'm using my body language, my hands, and also my lips in order to understand everything that this animal is communicating, right? So this is interesting because it means that this information has to come from multiple sources, and the receiver is responsible for integrating and decoding what those sources mean. We know a lot about how senders, which in the bird world tend to be the males, can vary in their signal production. Anyone know what this species is? House finch. Beautiful. Just such a great example of phenotypic diversity. So based on what fruits or vegetation this animal eats and the amount of carotenoids that are deposited in those that vegetation, depends on how much of that carotenoid deposition that male can place into his plumage. And females tend to prefer this very red male on average, because the signal conveys that this male might be a good forager, might be able to find the best food or is holding the territories with the best food, or might be able to produce the best food for its offspring and find and feed that offspring. So we know a lot now about how males can vary in their signal production, but the field didn't know as much about how females might vary in their sensory reception of these male signals. And so my PhD really focused on whether or not females can vary in their sensory processing and whether or not that can be tied to mate choice. Now, I can look around the room and see that some of you are wearing corrective lenses, some of you are not, right? And so I can already know that, yes, signal processing varies quite a bit among humans, right, within a single species. But that same phenomenon was just not as well studied within female avian species. The point I illustrate here to my students to really drive this picture home is that even if there's visual differences in how we process visual signals differently, our brain can impose those visual signals, and we can look at the same photograph and argue for days about whether or not this dress is blue or gold, or blue or black, or gold or white. I feel like there is, I always see gold and white. Does anyone else agree with me? You guys see black and blue? You see gold and white? Black and blue, see? I see gold and white. So that's my brain, based on the background of this photograph, imposing color which is a human-made construct, onto a photograph, right? So at the time of my PhD, when looking at the 100 most recent publications on mate choice or sexual selection, we see, again, what I've been saying is true, that of those top 100, 50% were focused on signal production in males. A mere 15% was focused on signal reception. And so I asked the question, does female multimodal sensory physiology affect her mate preferences, her mate choice? And just to give you a little illustration on why this might be important, let's take those male finches again. So we have male coloration here on the x-axis. We have a yellow male all the way up to a red male. And then we have a measure of female preference. Let's say we have an animal that can easily distinguish between these two or between these three males, right? She might rank them like this, where she follows the average trend in the population, where she prefers that most red male. This is an example of what on average should be occurring. But what happens if we have an individual animal that can no longer discriminate between these three different color morphs? And instead, her preference for any given male could be the same across these three individuals. Over time, this can lead to different types of evolutionary selection on the different morphs. The first one is an example of directional selection, where more males in the population are going to end up being more red because that's the one that's chosen more frequently. The second example is one of random mating, where any male has the chance to pass down his genes. And so that maintains that variation within the population. That's why studying female preferences are so important, because we can get different trajectories on where that population will be going. To study this question, I use a species of bird called the brown-headed cowbird. You guys familiar with those? Yeah. They're pretty notorious as a pest species because they have this interesting behavior where they parasitize different host species. And instead of building their own nest, they'll choose a nest of another nearby species. The female will lay her eggs and then leave and say, no parental responsibilities for me. Pretty advantageous for those cowbirds, right? I studied not that aspect of their behavior, although it's really interesting, but I studied the fact that the males use a multimodal courtship display in order to attract females, right? Males, previous to my work, were shown to vary in the intensity of that display. It could be a larger display or a smaller display. And males could also vary in the components of their song and change what types of song that they would sing back. So let me show you a little bit of what that looks like here. Here's a male on your left. Pretty fast. 1 second. Do one more time. Why not? 1 second song. What's really interesting is that the components of that song, it starts out sounding a little bit like water driplets. And then what you hear the best is probably that high frequency whistle at the end. But in between each of those acoustic elements is when he's doing those really rigorous visual displays. It's really tightly linked. Now, females, if they are willing to accept this male as a mate, will give a display that looks something like this. It's called a copulatory solicitation display. Pretty unambiguous, right? Yeah. We can measure how long she holds that posture to give us an indication of her preference for that particular male display. So more time means greater willingness to accept that male as a mate.
Dr. Ronald, are these two birds looking at each other, or what is their relationship to one another in this study?
Yeah, I'll show you. I'll get a little bit further into what I ended up doing with these displays. These particular individuals, no, but they are recordings of animals that I had in the lab. And so I would use this male as a playback to different females in the study. I'll show you what that playback design looks like. Good question. Okay. See if it comes up here. So first, in order to talk through the outline of this study, first I'm going to have to tell you a little bit about how the males vary in these two different signal types, just like how I told you about the finches that varied in their color. Then I need to map on female processing of auditory and visual signals that can match the different display types that I'm talking about, to determine how that female physiology affects her perception of male signals. And what I'm looking for here is really a significant statistical interaction between female preference and male display. That means female physiology is affecting her preference for a particular male display. So just keep that tucked in the back of your head for right now. To ultimately answer the question, does this influence preference? Okay. So first I'm going to start with the auditory signal here. Male cowbirds can vary quite a bit in their song types, and different males will have different repertoires of song. I'll play a few here. You guys heard the water droplets on that one. All right. And previous work had categorized these songs as either highly attractive, meaning more likely to elicit a female CSD, or less attractive. However, we didn't know what the specific part of the song that females found the most attractive in this particular species. And for us, just listening to it, I wouldn't be able to tell you, oh yeah, that one sounds like this and this one sounds like that, in order to be able to give you a prediction on this is an attractive song versus not. All right, so we had to take a closer look at a given song. And to do that, we split it up into two different components. First, this phrase one, those little glug glugs, those water droplet sounds. And then this final high frequency note at the end. What you're looking at here is what is called a spectrogram. So we have time on the x-axis and then frequency on the y. So higher is higher frequency. And then color denotes loudness. So you can see that this really yellow white is the loudest part of the song, which is why you're able to hear it the best. And we took different measurements across each of these components, measuring the duration, how long it was, how shifting of the frequency occurred, and also what that mean frequency was of each of those components. And then I could take all of those variables from each of these components of the song, the two glugs, and then this final phrase, and I collapsed it using a statistical method called a principal components analysis, which just lets me collapse all these variables down into a single one or two that helps me to understand the biggest impact on that component. Now, to characterize the visual display, I'm going to show you again what that first display looked like. And I would categorize this as pretty intense. Fully extended his wings. He dipped all the way down. He did a bill wipe at the end. But males can also display like this, just not as filled with gusto, right? There was less, it was overall shorter. There was hardly any wing extension. And so they can vary in how intense this display is as well. Overall, we took images across each of these different parts of the display, and then we could measure things like the width of the wing spread, the depth of that bow, overall display duration. And I did that same statistical method where I took all of those variables and collapsed it down into a single axis that I could use to describe display intensity. Okay, here's that promised playback design, right? I have a female in an arena right here. I have an LCD monitor showing that male display. It's going to come on this display here in a few seconds. There you see it appear. And then you see that female give a CNC to the video monitor. So this design of, it took me a long time to get this to work. The females ended up needing estrogen implants in order to mimic the same types of hormones that would be similar to what would be occurring circulating in the wild. But this allowed me complete control over what types of males this female was viewing, right? So I could vary the songs with the visual intensity and a full factorial design and ask females questions. What is your preference? Overall, measuring that display duration.
Looking at the male display, you think the lesser display is less interest in that particular female? They could display multiple amplitudes of their display, but they're attracted to certain females more and they put a larger display. Is it only that they have one display and the female makes a choice, or is he making a choice as to how to display ahead of time?
That's a great question. The question, kind of paraphrased, if I understood it correctly, was that can males decide how intense to display depending on the receiver? And the answer is yes. So similar with the songs, they can choose which songs they want to sing to a given receiver. Interestingly, in this species, they found that males will tend to give these larger displays to males as an aggressive display, and that females actually tend to prefer the lower intensity displays. And that was work not published by us, but yes, they can absolutely vary that display intensity. Right. So now I needed to map on the measures of female auditory physiology that would help her to be able to distinguish these different types of traits. First one I'm going to talk about is auditory sensitivity, the ability to resolve low intensity sounds. And this might be particularly important for those first few notes that are sung at lower amplitudes. And those have been shown to be particularly important for female choice in this species. Finally, the other thing that we measured was auditory temporal resolution, or the ability to resolve fast changes in sound. So are you able to distinguish each of these as a separate beat? And at what point does it no longer sound like separate beats, but instead just a single note? And that might be particularly important in this species because of how rapidly they sing those songs, right? And go through those different elements. So how do we test this? This is something that I started within my PhD and have continued here at Hope. And we use a test called auditory evoked potentials. And you might be familiar with these. It's the common newborn hearing screening test that's given. I've also had a ABR done when I've gone into an audiologist to assess my own hearing. So in newborns or in adults that can follow instructions, we can close our eyes, be really still, just skin electrodes on, and then different types of sounds are played back. And then we can record at the level of the auditory nerve that's responding to those playbacks, how is that auditory system functioning, right? Now with our birds, they don't tend to follow instructions when I tell them to do things. So I have to help a little bit. We sedate them with a mixture of ketamine and some midazolam so that they're sedated. But that auditory nerve doesn't stop responding even though they're asleep. And so once they're sedated, we can play back different types of sounds and I can record what is called an auditory brainstem response. Looks a little bit like this. You see a peak up and then down. And what we're really interested in measuring is how soon does this respond? How large of a response is this? And then we can change parameters of this sound. This is played back at 72 decibels, which is pretty loud. But if I change how soft that sound is played back, eventually we see that ABR goes away. And that's the point at which the bird physiologically can no longer distinguish that sound from background. And that's what we call the threshold, right? The lowest level sound that the animal can resolve. Okay. I'll be showing a lot of these different types of graphs throughout my talk because it's a methodology that the lab does frequently. Okay, so what do we look at in terms of auditory physiology? We looked at that threshold level, the lowest level of sound that evokes an ABR to give us an indication of auditory sensitivity. And here it's important to know that lower means better hearing, right? If you're able to resolve a lower intensity sound, you have better hearing, right? Right. And then I also measured something called auditory filter width. This is a physical property of the cochlea that allows for temporal processing. Here, I just want you to know that wider auditory filters allows for higher temporal resolution, meaning you're better able to pick out single notes as unique and distinguishable. Okay. So what did I expect with these two auditory traits and then how this would map on to female preference? I expected that animals with higher auditory sensitivity, better hearing, would tend to have a preference, a stronger preference as that frequency of sound in that first component of the male song went up. And I expected that animals with lower auditory sensitivity wouldn't necessarily show a preference across this, okay? This is where that statistical interaction comes from. I'm expecting different traits of the female to indicate that she's gonna prefer different types of male components, right? And then with temporal variation, I expected that animals with a better ability to resolve temporal information would prefer shorter songs, meaning they were able to better distinguish really quick beats compared to animals that had lower auditory temporal resolution. So what I'm going to show here is a significant interaction effect, yes, for our CSD duration, our dependent variable of how long is that female display. On the x-axis, I have phrase 2 duration. So here, lower on the axis means shorter, higher means longer. And I've divided up the females into different quartiles. So here's our group of females with the lowest auditory temporal resolution. And then I have mid-low, mid-high, and then here are our animals with this highest auditory temporal resolution. And what I really want you to take away is look at the change in the best fit line as we pull away those panels, such that animals with the lowest auditory temporal resolution tended to prefer the longest songs. Animals with the highest tended to prefer the shortest songs, matching the predictions that I had. Okay. Now let's talk about visual. I also looked at visual temporal resolution, the ability to track changes in a visual signal as it's changing over time. This might be particularly important in terms of motion-based signaling, like what the male cowbird does when he's making that motion-based display. In addition, I also looked at visual spatial resolution, so the ability to distinguish 2 different points as unique in space. And the parameter I use for visual physiology is very similar. I use visual evoked potentials. And what this does is instead of having a sound at a frequency or a decibel level, is instead we could vary the flashes of light that were presented to the sedated animal with an electrode placed right on the cornea, right? And so this light could vary in the frequency of how quickly it flashed, right? to the point at which we could find the flicker fusion frequency, the frequency at which a pulsing light is no longer seen as flashing. So in here, these lights are, as humans, our flicker fusion frequency is about 60 hertz or so. And these lights are flashing probably right above that, these little LED lights. So we don't see this light stimulus as flashing. But birds have a higher flicker fusion frequency. So if we bring them into the lab, which is what we do, it looks like a strobe light in here, right? Because they're able to resolve and see each of those flashes of light. And it's not perceived as a steady beam. So to illustrate this, if you don't like flashing things, you might want to close your eyes, right? So starts off slow and eventually flashes so quickly that we can no longer see that stimulus as flashing anymore, right? So birds with a higher flicker fusion frequency would have higher temporal visual resolution. Now, for spatial resolution, we looked at the number of cones, the primary photoreceptor that's responsible for detecting color on the retinas of these animals, right? And higher density means higher spatial resolution. So higher number of cones, better able to resolve those visual signals. Interestingly, I just love showing this slide because this is a retina that's placed on a slide and you're just viewing it as a light image on a microscope. Animals, birds, not humans though, sometimes have these pigmented oil droplets within the middle of their cones, and that filters the light before it enters the part of the cell that responds to that visual signal, and it almost looks like stained glass. Interestingly, these are pigmented with the same carotenoids that the animals get from their diets, suggesting a link between how well an animal feeds itself to how well it sees to looking at visual signals. Love that. Okay, so what were the predicted interactions that I had in this part of it? The visual traits that I was looking at was visual spatial resolution and visual temporal resolution. And for both of these, I expected that animals with better abilities to resolve these visual traits would show stronger preferences than animals with lower visual abilities. And so what did I find? I'm going to show a very similar type of graph here, where now instead of song length, I'm going to show you visual display intensity. And I've split these into different quartiles. So now blue here is the lowest visual temporal resolution. And then this panel down here is our highest performing animals. And what we see as I pull this away, again, is a change in the slope of that line, meaning that females that had this highest visual temporal resolution actually ended preferring these lower intensity displays. And females that were perhaps not able to resolve those movement-based displays tended to prefer the ones that were highly intense. Okay, so this is the first time that we've been able to map physiology to a particular behavior across two different sensory modes to be able to understand female choice. And so just going back to summarize all of these predicted interactions, we did find that there was support for this auditory temporal resolution, where females with higher abilities to resolve would prefer those shorter sounds. Visual temporal resolution, I also found a relationship there where females with higher visual temporal resolution tended to prefer display intensities that were short. Here with auditory sensitivity, actually didn't find a significant interaction, but I did find a main effect where females that had higher sensitivity just tended to give longer CSDs overall. We think that might just be related to motivation, motivation to mate, perhaps, compared to animals with lower auditory sensitivity. And then finally here, even though I spent years counting cone densities, because we haven't developed AI to be able to do this for me, it was me and several students. There was no relationship between cone density and display intensity on female preference. I will say here that this did not match my original prediction, but I still wanted to give myself a check mark because I did find that significant interaction effect that I was hoping for. Just wanted to give myself some motivation. So overall, what does this mean, right? What does this mean? It means that females vary. Females vary in their multimodal sensory processing, which seems simple, right? But in the animal world had not been shown before. And it suggests that not every single male is going to be attractive to any given female. Females and her sensory physiology changes the types of males that she has preference to. And so it's important to improve communication across both sensory modes, right? So there are lots of research groups out there that examine one modality over the other, but by doing so, you're losing an entire source of information. And so it's important that we still consider multimodal communication when we're talking about sensory reception. And this is particularly important as we transition into talking about what the lab is doing now in terms of studying urbanization, right? So this Times Square, and we can almost hear this picture, right? I'm showing you a visual stimulus, right? I'm showing you light pollution, but we can also hear the noise in this picture almost, right? So I don't have to convince you that urbanization spans multiple modalities when we're talking about pollution. And this is only expected to increase, right? So urban land cover is projected to triple from what it was in 2000 to 2030, coming up here soon. And as we plot where humans were living in the 1950s, a greater proportion of the global population was living in what was considered rural areas. By 2009, those two groups were considered equal. And now the amount of people living in urban areas is skyrocketing and only expected to increase. If we look at this in a pie chart format, we can see that in the 1950s, greater percentage living in rural, all the way up to the projected where 68% of all humans will be living in urban areas by 2050. So this is not going away, right? This is not a problem that, and maybe it's not a problem. There are benefits, right? This is worldwide, yes, yeah. Worldwide population, right? Lots of benefits to urbanization, but challenges that come with that for our animals that are having to live alongside us. All right, so the question that my lab is focused on right now is how does this increased urbanization affect sensory receivers? So still looking at that mechanism of animal physiology tied to behavior. And the two ways that we do this, oh, that's another slide. So I want to talk first about what we know about signal senders, right? We know that urban noise can inhibit a bird's ability to communicate through song. So these are two graphs of sound that were recorded by my two student researchers, Sarah and Eliza, from around the Holland area. And in rural areas, you can see that that noise in terms of amplitude is a lot lower compared to in a more urban area like downtown 7th Street right here, right? And these are the songs that are being played back. You can see how that noise tends to disrupt the ability of a receiver to hear that sound. On a more global scale, we can see that animals are adapting to this, right? So in order to overcome this low frequency traffic noise that is dominating our urban landscapes, birds will tend to increase the frequency of their song, sing higher, get out of that noise. And they'll also sing louder to try to overcome that loud noise that's blocking that signal. So this is in an urban area. You can see that minimum frequency shift up compared to the same species that's living in a forested habitat. The pandemic allowed a unique opportunity where noise suddenly and drastically decreased. If you guys ever get the chance, I highly recommend watching. There's a documentary that's called The Year the Earth Changed, and it's all about David Attenborough, of course. And it's all about how the COVID pandemic and the arthropods that resulted from that changed how animals communicate. And some of the greatest work is coming from the Derryberry Lab from the University of Tennessee. They have a long-term study of white-crowned sparrows in the San Francisco Bay Area. And you can see that this population of white-crowned sparrows, this background noise, see rural areas have much lower background noise compared to the urban areas in the Bay Area, but that during that COVID shutdown, that basically was eliminated. And vocal performance, the differences in vocal performers was also eliminated, meaning that urban birds that used to have to sing at a lower performance rate than their rural counterparts were able to quickly adjust, even within a few months, to be able to sing at a level where their communication was more effective. Right? So things can change. Birds can adapt and adjust their behavior very quickly. So the way that the lab studies this is with two different approaches. One is through the use of observational studies, where I go out and I sample what is already existing. Right? I don't impose an experimental manipulation of my own. I go out and sample and see what's occurring in the area that's around us. The other, much harder to do, is an experimental manipulation. So me being able to control within a laboratory setting the exposures that animals receive that help to mimic and help us to predict and understand a cause-effect relationship. So I'm going to talk first about the observational studies, right? So for this work, the lab is primarily using the house sparrow, right? If you are birders in here, perhaps you have a love-hate relationship with the house sparrow, especially if you have bluebird boxes or purple martins. They're invasive species from Europe. They like to come in and knock out those native resident birds. They can be aggressive when setting up those nest sites, and so they can cause a lot of damage. That means they're a great organism for me, right? I can take any off your hands that you need. And they're particularly a great study system for studying the process of urbanization because they are anywhere humans live, right? So they span this rural to urban gradient, kind of setting up an experiment in and of itself in which I can go out and test how do different populations of house sparrows respond. Okay. So the first work that I'm going to show is data collected from my student Linda. She's graduating this year and has been accepted to MSU in Illinois for veterinary school. So I'm sad to lose her. That's the problem of working with a four-year institution, right? Right. They just leave. Her work and question was, if we go out into the Holland area and sample from different populations that we're able to denote as more urban versus less urban along this urbanization gradient, does hearing sensitivity change across these house sparrows? And so these are our different points. Linda's prediction was that animals in more urban locations may have decreased hearing sensitivity, meaning higher thresholds, right? If you recall that. So she ran 60 ABRs in the summer of 2021, to which we were able to collect those data and answer this question. And the answer is complicated, always. Despite our efforts of collecting data within a single season, right? We collected data from March until July, breeding season. We still found a date effect, meaning animals' hearing was changing from March until July. It's not necessarily unexpected, but wasn't something that we were setting out to test. We also found a sex effect. Males and females hear differently. Also not necessarily unexpected. That's why we study both males and females. But what we were particularly excited about was that we did indeed find an effect of our urbanization level in addition to all of these things. So let me orient you to this, this graph here. On this side, we're going to have our rural animals. On this side, we're going to have our urban animals. And then within each of those, I'm going to have to plot males and females because they were different, right? And then we're going to have two different seasons. Early breeding season, which we're going to call spring, and then later breeding season that we're going to call summer. And so here's spring. Here's spring. In rural habitats, kind of what we expect. Females have better hearing. Remember, lower threshold is better compared to males. Females are the ones that are doing the mate choice decisions. They're the ones having to really, really listen to see where those males are and locate and find those potential mates. In urban areas, however, we see that males and females don't differ. Okay? This is only occurring in the rural areas. And then when we start mapping on summer, we see that by the time summer hits, our rural females' auditory physiology changes to look more like males again, right? They're coming out of that breeding season. They don't have to pay as much attention to those male signals anymore. They're off feeding their offspring. But that opposite trend is true in urban areas, where now it almost seems like female communication or having to listen in is almost delayed in our urban birds. They're having to wait longer to find and perhaps participate in those mating decisions. And we're seeing an interesting effect where male hearing is also changing within those populations too. Complicated, but interesting. When I shared this with my PhD advisor, he was giddy with excitement. Scientists just love complicated things, I suppose. Complicated stories. Okay. So now moving into what are those really, really necessary experimental manipulations? Part of the talk that you probably came to hear, right? This collaborative project that is ongoing between myself and Dr. Gonzalez-Pech. So I have to start you off by saying that we now know that air pollution alone, a single factor, is responsible for over 5.5 million premature human deaths worldwide across 188 countries. So this study published in 2015, huge meta-analysis, huge review that was able to look at this effect within humans, right? A year ago today, well, almost today, it was suggested by the World Health Organization that 99% of humans breathe unhealthy air, right? And so let's take a closer look at what is going on here.
Air pollution, it lurks all around us, preying on the young and old. It slips unnoticed past your defenses and assaults your lungs, heart, and brain. Its deadliest weapons are invisible, poisonous particles that can be as small as a molecule. With every breath, they break through your lungs' protective barriers. There, they trigger inflammation as your system tries desperately to fight back. But these tiny intruders penetrate those defenses, lodging toxic compounds even deeper, sowing the seeds of cancer. They slide straight into your bloodstream, sabotaging your entire body. They inflame and constrict your blood vessels, increasing blood pressure until, one day, causing a stroke. The particle prowlers dislodge fatty plaque that may have built up from your diet. They weave clots that can be deadly blockers of blood flow to your heart or brain. With each new case of heart attack, stroke, lung disease, or cancer, air pollution adds another life to its hit list. Let's stop this invisible killer.
Huge effect. So that video mentioned that there are a couple of different components of air pollution. The gases that are comprised within the pollution that's given off, but also the particles. And it's particularly these particles that can cause the greatest damage, although I would argue that we know the least about them, right? The lowest size that governments have categorized these particulate matter is PM 2.5. So that means particulate matter, PM 2.5, is in microns, right? So if we look at the strand of human hair or a grain of fine beach sand from our Michigan beaches, we see that PM 10 are these blue dots that can go around the width of this human hair. PM 2.5 microns or less are the red dots that go around each one of these little dots, just to give you an idea of scale. And to me, that's still huge because we're measuring at the scale of microns, right? In reality, we know so little about even smaller particles that are at the nano size scale. We'll play this one really quickly here. That last little phrase, that dramatic music said, we don't even have a good way to measure nanoparticles in air, right? And all the laws and protections that we have in place were at a scale in the microns, significantly larger than what we're beginning to realize causes the damaging effects. And so this is where my colleague comes in to play here. Dr. Gonzalez-Pech came to me with this idea when we first started. We started the same year in 2009. And she came to me with this crazy idea. And I was looking for a reason to help avian populations and interested in really, really broad topics. And Dr. Gonzalez-Pech's lab has the ability to do what that slide said that we can't do: measure nanoparticles. And so within her lab, she's able to not only measure them, but also produce them, right? Produce nanoparticles that are similar to what we find in air pollution, and then also quantify their existence, right? Based on, this is a image of, this is at 500 nanometer scale. And so these are actually clusters. This is not the nanoparticle, but they are actually the smaller components within those that are agglomerated together. But then you can see in here this size, right? So, and where those particles are clustering. That little video said that these nanoparticles are small enough to bypass our blood-gas barrier, our blood-brain barrier. And when Natalia was coming to me with this idea, she brought this paper with her that said that magnetite pollution nanoparticles had been found in deceased human brain tissue, right? Published in one of the leading journals that we could, as scientists, hope to get published in the proceedings of the National Academy of Sciences. This is a huge deal, right? Even just this week, Wednesday, April 5th of 2023, our governments are trying, excuse me, to make calls and proposals to enact stricter air pollution targets to help tackle this link that has been found between dementia and air pollution. So in addition to heart disease, cancers, we're also finding links to neurodegenerative diseases as well. All right. And of course, me, right? I love humans more than anything, but I also care a lot about our avian species and our wildlife, right? And although we know quite a bit now, data is kind of unrefutable that this air pollution is causing huge health impacts for humans. What about the implications for wildlife? At the beginning of that video, air pollution was not among one of those listed factors for what might be causing sudden, within a single generation, decline in species. And so perhaps this is one piece of the pie, right? So we still do not understand how the same pollution affects wildlife, particularly vulnerable avian species. Why do I say particularly vulnerable? Have you ever heard the phrase canary in the coal mine, right? There's a reason behind that, right? Coal miners used to take a canary down into the coal mine to test the air quality before even they could detect that it was not of good quality. If the bird passed out and fell off the perch, the coal miners would know, oh, it's not safe, there's not enough oxygen. They would carry that bird back up. Sometimes you could reuse the same bird over and over again. It woke back up. And then to give an indication of air quality, right? And so when we were writing our grant, this was included in the title of our grant, and was also the huge justification for why birds might be an appropriate model species as a sentinel species for being able to detect air pollution concerns. So unique to avian species, they have a unidirectional airflow, right? They don't inhale and then exhale. It goes through the air pathway, goes unidirectionally through the respiratory system. And this makes them particularly vulnerable to airborne contaminants and also useful models to study this. Right. We had to choose what type of nanoparticle we wanted to examine, and we settled on iron oxide nanoparticles because they're one of the most abundant in air pollution caused from urbanization, in particular from iron and steel industries. It also they can also be produced from non-exhaust emissions, like just the friction of your brakes on your tires can produce these nanoparticles. But it is these iron and steel industries that contribute up to about 90% of these iron oxide nanoparticles into the environment. The paper that convinced me was this one. All right, I needed some convincing. And I came across this one. Air pollution is associated with brainstem auditory nuclei pathology and delayed auditory brainstem evoked potentials, right? That's my wheelhouse, right? That's the methodology that the lab does. And it gave me a aha, like, wow moment. Because if we can quickly and easily test with an auditory test downstream consequences, even in species like our birds, this might be even a better reason for using these animals as a sentinel species, right? We're expecting changes in hearing. And so this paper kind of led the production of a grant that was eventually awarded. We submitted it last January. It was awarded last September. And these were the three main points, the three objectives that we wanted to address within the grant. First of all, to identify whether exposure to these nanoparticles decreases hearing sensitivity, right? Coming off of this paper, which is what they found. This was in humans, so in children, from areas with lower and greater air pollution. It's also been replicated in other model organisms like canine species. The second aim that we wanted to address was, is there a link between hearing and behavior, right? I'm a sensory ecologist. That's what I do. I link physiology and behavior. And in particular, we're looking at the ability of these animals to respond to a predator and also to detect food and find food, right? Survival, right? And then finally, to localize and quantify the accumulation of these iron oxide nanoparticles in relevant organ tissues, using, again, this house sparrow as a model system. So this is kind of the experimental design of what we did. Initially, this was back in the summer of 2021. We would bring the animals in. We would run a hearing test on all animals. We would examine their behavior to a predator response. Then we would either have a control stimulus where they were just exposed to saline solution, or we would expose them to the nanoparticles that were synthesized in Dr. Gonzalez-Pech's lab as our treatment stimulus. And then we would repeat. Did behavior change? Did the hearing change? And then at the end of the day, where did this iron bioaccumulate, if any? And so I can't go further without talking about the work of my students. I've already talked a little bit about Sarah and Linda here, but I have other students as well that have contributed so much to this project. So we have Olivia and Lindsay, Peyton and Molly and Jacob. That's me in the middle there. And then Andrew is the undergraduate student that is from the Ghost lab that helps us synthesize all these nanoparticles that we end up exposing the animals with. All right, so first I have to talk about that synthesis. What does Andrew do here, right? He helps by creating these nanoparticulates within a solution that we can then use through a nebulizer to expose the animals to these inhalation studies. We do a process called sonification, or which basically spins and breaks up those agglomerated nanoparticles to be of smaller size. And what that also does is it helps to maintain the stability of when we can use those nanoparticles. You can see that after this sonication, we don't really see a significant change in nanoparticle size until about week 7. We tend to use them by week 4, right, to give ourselves plenty of wiggle room before we use them in our exposure chambers. The data for submitting this NSF proposal was using child nebulizers purchased from Amazon. Thanks, Jeff. And then with these are anesthesia chambers for putting mice to sleep in research studies that use mice. And so we hooked up. My students helped to hook up these nebulizers. Our solution would go into here, and we would force that air into this chamber where the bird would be. And you can see that this exposure would change that filter paper that's lining. You can almost see the bird outline here, to this exposure. All right. Today, I'm going to focus on what we found in terms of hearing and then organ quantification for the purposes of time. This is the data that I submitted to NSF that was the most convincing piece to me. So remember that I said that we tested all the animals on their hearing before exposure. And then we had two groups, one that were just given a control of saline, and then in orange are the ones that were given nanoparticles. We see that the animals that from the before trial, and then also the control animals in blue really overlap, right? So these are the different frequencies that we test in our hearing study. Remember, threshold here, lower is better. So our birds are hearing best around this 2 to 3 kilohertz range. And then when I plot on our nanoparticle exposed birds, we see a huge change in hearing sensitivity, right? Where our nanoparticle exposed birds had huge increases in that threshold, meaning very, very decreased hearing sensitivity. This was very, very convincing to the research team, and made a case for why we needed to do these studies properly with this pilot data, right? We also, I haven't yet shown you a plot of what happens with latency or how quickly that auditory nerve responds. Here we have, again, just another indication of hearing changes. So we have the different frequencies plotted above here. 500 to 6,000 is what we usually test. And we see that those latencies of that response are also delayed, very similar to what we're seeing from reports in human children from polluted areas. Okay. So now going into organ quantification, at the end of the day, we do euthanize all of our birds. The government protocols that I have, they do not want me releasing house sparrows back into the wild anyway. And so using these animals as efficiently as I can to address a really crucial problem is the way to go. So the animals are euthanized. And then we use a machine called an ICP. It's kind of like a very fancy mass spectrometer to quantify the amount of iron in each of the organs. And this is what we found in terms of that iron concentration. Now, if I had my choice, there would be huge differences, right, in these iron nanoparticle exposed birds compared to the control. But you can see that there's really big variation, right? Perhaps it's due to the sample size. These were just 4 birds within each group. And so we've been investigating why this might be the case in our future endeavors. One of the reasons or thoughts that we had while we've been investigating this is perhaps it's the length of time in the lab, right? The birds that we used in this first study had spent quite a bit of time in the lab, about 6 months. They had been in the aviary that we have on campus. And so perhaps it's the recently caught animals would have less variability than our birds that have been in the lab for a while. And so we did what scientists do. We asked the question, is this the case? Does being an older bird mean that your iron levels are going to change compared to being a newly exposed bird out in the wild, exposed to the elements? Still tons of variation here. Error bars that are really, really large. From a lab population of mice that we examined, we see that those mice actually tended to have lower levels of iron overall. and overall less variability. So perhaps there is some indication that a wild-caught species is just harder to quantify these levels in, whereas a completely lab-raised animal, you're going to have more control over those environments. All right. This past summer, we changed this process by a little bit. I had a student that was particularly interested in this link between cognition and nanoparticle exposure. So we started doing some foraging trials where the animals were having to do a puzzle to find food. But the overall design was the same. And even with a larger group of animals, 15 now, rather than 4, we're still seeing large amounts of variation within our nanoparticle birds and our control birds, suggesting that there's just inherent variability in the animals, probably the case, or perhaps that our method of quantifying needs to be adjusted, or where these animals are coming from needs to be factored in. And so we ran that test. What happens if we look at where these animals originated and the organ that was collected on these iron levels, and we found a significant interaction. You see the red bars here are our urban considered animals, and overall they tended to have a little bit higher iron concentration, perhaps just from the environmental exposure that they are getting in the wild. And so future work really needs to consider where this location of the animal is coming from when we are choosing our animals for this particular study. For our ABRs from this larger data set, larger number of animals, right? You get more variability. So here, now instead of just having a treatment effect, we are seeing that the sexes vary. That's not new information for you. And across the frequencies also changes how they hear. Now, with males, it followed the predicted trends where ABR amplitude, so here now larger amplitude is better hearing. So our control animals still tended to have larger amplitudes compared to our exposed animals. But with females, it became a little messier, where now we, instead of seeing this clear pattern, it looks like that audiogram has even shifted so that they become worse at lower frequencies, but perhaps better at higher frequencies, suggesting that exposure may affect the sexes differently, and that this can result in differential hearing outcomes. Okay. All right, so what is next, right? With this or almost $500,000 grant, what are we doing? We are thankfully updating from Jeff Bezos nebulizers to equipment that allows us to standardize and quantify nanoparticle exposure in a true way. So this equipment was installed in February, and we're still in the process of testing and going through the procedures to make sure that we are exposing the animals to a known concentration of these nanoparticles that we want to do. We have one setup that is for control animals, and then another setup that is for our exposed animals. Okay. We're also continuing to explore this relationship between physiology and behavior. I'll say behavior is one of the hardest things to measure in an animal, right? It's easy for me to sedate an animal and collect an auditory response. It is much more challenging to understand why that animal is behaving the way that it's due and the variability that can occur there. So we still have quite a bit to do in order to really understand these puzzle studies and these anti-predator studies, but it will be ongoing. So I hope I've left you with Convincing that house sparrows can serve as this new model for a canary in a coal mine as the lab continues to use observational and experimental approaches to investigate communication within an avian species. And so all of this with the hope that our data can be used to help increase conservation efforts and be the first to look at this impact of air pollution on wildlife communities. So with that, I'd like to thank the funders and my students again, my collaborators. And if we have time, I'd love to take questions. Yep. Thank you.
Am I correct in assuming that there are fewer nanoparticles in a rural area than urban?
I think
Why I'm asking you that is I'm wondering how you know how much to expose the birds to.
right. Yeah, our chosen levels, I will say that this grant is over a 3-year time period. And so the first thing that we are doing is a very high level acute exposure. So it's not environmentally relevant, but it is related to levels that have been chosen in mammalian species to try to stay within the literature framework for being able to say, like, this is what has been found in mammals. Now let's test this in birds. Later on, in the later half of the grant, we're planning on doing a more chronic exposure where we do this environmentally simulated amount for a longer period of time. And that will be more related to what is actually occurring in the wild. Yeah. But I think to answer your question, are there more nanoparticles in rural versus urban? I'm going to do the classic, it depends. Perhaps with this particular iron oxide nanoparticle, we could say that closer to these iron and steel factories, that might be in urban areas, yes. But if we were to look at another particulate, that might change. Yeah.
My question is, do you know how old the birds are? Like I'm thinking, you were talking about the variability, like
Right.
maybe some have been exposed to the environment, whatever's in the environment longer than other birds.
Right.
And that could maybe
That's
be a reason why some of your variability is happening.
Yep, that's a great question. We are able to tell a bird that was hatched this breeding season. We try to avoid those animals. So we try to collect animals that are at least in their second breeding cycle, but telling apart a 2nd-year bird from a 4-year bird, nope, can't tell. So it very well could be. But kind of also outside of our knowledge base, at least at this point. Yeah.
Quickly, at the very beginning, we talked about the bird loss. Is that numbers that you gave, those are worldwide numbers, or are they US numbers? Are they Michigan numbers?
North American, so including Canada, the United States, and Mexico.
Okay,
America.
or the 30%
Yeah.
loss.
Yeah.
As you're doing your studies, and since pollution is a major factor in all of your research here, is Holland, Michigan the best place, since we have Lake Michigan giving us cleaner air than perhaps California? Is that factored into your research?
Yeah. That's one of the most common questions that I'm asked is Holland, Michigan urban? And my answer is not really. However, we're still seeing effects, right? So we're still seeing that a bird caught in downtown off of 7th Street is different from an animal that's caught closer to Byron Center. And so, more money to get me to further locations so that I can ask this at a more appropriate, broader scale. So next steps. Yep. I love it. I'm hopeful that, Grand Rapids is my next targeted area. Just take that step a little bit closer to a more urban area. Yeah.
Yeah, I was wondering if I'm understanding this right. The loss of birds is due to, there's less breeding of birds due to the pollution affecting their song and display and hearing, or are birds basically dying more because of the nanoparticles, or is it a combination of both of those?
Yeah, I would say both. So when you say pollution, I encompass things like air pollution, which is where my mind goes to, but also noise pollution and light pollution. So we know that bird communication is negatively impacted because of noise pollution. Can I say that about nanoparticles? I can't say that senders are changing their song because of air pollution. That's yet to be studied, but we can say that receivers are starting to show decreased hearing when they're exposed to these pollutants. And so, these animals rely on sending and receipt of these signals in order to find and, attract a mate. And so if that is more challenging, then yes, it could lead to either not finding a mate, not passing on genes, and lower bird populations. Yeah. Yeah.
Any other questions here?
Question from online that are curious about your bird capturing techniques.
Oh,
Did you or did your students do this?
I almost put in a picture of the trap because I was like, maybe I can recruit people that don't want house sparrows in their yards. So I tend to use a baited seed trap. House sparrows are another great species to use because they eat seeds. And so those types of animals are much easier to maintain in the lab. I don't have to provide them fresh insects, on a daily basis. And so it's a repeating trap. And if you just Google housesparrowtrap.net, you can get one shipped to you very, very quickly. And it's a repeating trap. So you bait it for a while, the animal gets used to coming in. And then once they come in and the trap is set, they are live captured, and it can repeat multiple times. So I could catch up to, it can hold quite a few. Yeah.
Did you do this with cowbirds as well? Same trap?
For the cowbirds, we did use a trap. It was a much larger trap. It was actually a walk-in trap that was about, in America, we use everything but the metric system to measure, right? So here, these two tables that I could walk in, and there was a funnel. And I actually worked with the USDA on one of their airport bases in Ohio to do a lot of that trapping. Yeah. Yeah.
It's large number of birds that die. Have there been any studies on what particular kinds of things are most lethal to them? West Nile, for example, or glass windows, or?
I would say domesticated cats that are not brought inside would be far and away the biggest killer of our wildlife. Yeah. Yes. Yes, I would.
Just a comment on the air quality.
Mhm.
We have Louis Padnos. They process thousands of tons of iron
Yep.
every day.
Yep.
And they have been known to produce plumes of dust particles and
Yep.
things. So I would think compared with a lot of urban areas, we have a very high amount of iron oxide nanoparticles
Yep.
near the Padnos works. I don't know how far that would extend, but Lake Michigan air is not going to save us from those iron nanoparticles.
I have considered including that in my response to that question. Often the results that are closest to publication are the ones where we did a song propagation study in different areas that I'm asked that. But yeah, especially with this air quality, I think you might be onto something there where I can tie that. Yeah. Yeah.
I have a question. In the case of laboratory mice, they are bred, they're essentially sterile. They're genetically identical one to
Yep.
another. Do you wish you were able to acquire birds of that clean history?
My postdoctorate was in a neuroscience lab where I was able to work with these lab bred mice. And you see where I ended up studying wild caught birds again. My questions are, I want to be able to have realistic implications for the answers that I'm finding. And so, yes, there's variability, but that's also real life. And so I think I would prefer to work with those wild caught animals. That variability interests me rather than makes me throw up my hands.
So you're really not surprised that your results show variability?
No, not really. Yeah.
It's 2:30, so it's theoretically we're done. Are there any other questions? Well, I'd like to thank you a whole bunch for this.
Absolutely.
I'm blown away by the level of science that happens right here in little old Holland, Michigan.
Yep.
It's spectacular.
Pretty incredible.
So thank you all for coming. Thank you
Thank
all,
you.
all of you that are online, and we'll see you for the next session.